Nitya, do you want to just start by answering the two questions?
Yeah, I think I will. So the first question is, could dopamine or neuropeptide be a global error signal which may modulate the local synaptic learning? Absolutely. So this is an example of kind of a non-local synaptic learning rules. And it is possible, but it's rather limited. Why is it limited? Is because only one of very few signals can be communicated to all neurons non-selectively, because these signals are usually based on long-range diffusion. It's just one signal. It usually is communicated on with a slow time scale, and it could work for something like maybe reinforcement learning algorithm, but it would be hard to use for something like backpropagation, because there, remember, the synaptic errors have to be multiplied by different synaptic weights for each individual synapse. And so that is very hard to accomplish with this kind of broadcast signal. So yes, you're absolutely right. This is an example of non-local signaling, but no, it's not specific enough to be used in a learning rule like this, okay? The second question is, does this online learning algorithm has anything related to recursive least square? Since for the offline version, the form goes also similar to the solution of least square. I can't answer this off the top of my head. It's not recursive least square in its full form, but maybe conceptually this is related. I don't know. I haven't been able to implement the recursive least square with local learning rules, so I actually don't know the answer. Okay. All right. So let's go back to the experimental test of the prediction that the synaptic weight vector is given by the top eigenvector of the input covariance matrix. And I just told you how we're going to get the W from connectomics, but how are we going to get the activity vectors? Well, for this same system, it is possible, because it's transparent and because it's genetically accessible, to measure activity of all the ORANs at once in response to the presentation of different orders, as was done in the Samuel lab, by genetically expressing a calcium indicator in each of the ORANs. And remember, calcium indicator responds to the neuronal activity, which raises the concentration of calcium, which in turn changes the optical properties of the calcium indicator that you can read out with your microscope optically. And so that's how this response was measured. These numbers here are the names of the ORNs in the fly larva. They're identifiable from animal to animal. And so you can record this kind of responses, a response matrix for different orders, how activity increases in various neurons. And then that gives you the data matrix X, the single value decomposition of which, or the eigenvector of the Gramian of which, X transpose, will give you the top eigenvector. So we just did PCA on that activity matrix, and this is what we got. So the green line is the PCA result, and the blue line is weights from connectomics. And as you can see, there is not a perfect, but a reasonable agreement between the profile of the synaptic weights obtained in two ways. And so this suggests that our prediction is not that far off of what is actually happening in the system. Now, at this point, I should also say that I don't want to oversell this result. There are many caveats here. For example, the data set of orders that was used here is not really a natural order ensemble. Of course, we don't even know what the natural order ensemble is. It just was chosen to be as broad as possible so that it stimulates all the receptors. But we don't know if it corresponds to what the larva really sees. And so if we believe that the synaptic weights adjust to what they see in nature, I think that there is a question whether this is the right data set. And my answer is, well, if we didn't get an agreement, then we could always blame it on that. But since we got some kind of match, it's unlikely that this happened by chance. And it probably does reflect the correlations that are present in the responses of ORNs to different chemicals. So we probably captured something real, even though our data set to probe the system was not perfect. The second caveat is that I oversimplified the circuit quite a bit. In addition to this one interneuron, there are several others also connected to the ORNs, and the W's that those neurons have are not actually do not actually correspond to second and third principal component. And the reason for that, there are multiple reasons. Of course, one could be, again, this data set is not perfect. But the second one is that neurons here are not really linear devices. Okay, they don't just compute the weighted sum of the inputs, they also rectify the sum of the inputs. And so because of that nonlinear computation, PCA is not a perfect way to describe what the network does as a whole. It's just a linear approximation. And so that could account for a mismatch. Are there any questions? Okay, if there are no questions, let's move on to the next experimental test. So remember, we also made a prediction about the learning rate dependence, which decreases with time because the sum of activity of the neuron grows and you divide by bigger and bigger number, and so the learning rate gets smaller and smaller and smaller. And so this can actually be tested, and it has been tested even before our theory was proposed. We didn't know about it. But here is the experiment. And this is the long-term potentiation that I mentioned before. Just to quickly review what this is, experimentalists prepare a slice from a rat brain. And in that slice, they manage to stimulate two neurons that are connected by a synapse. And first, they measure the strength of that synapse by letting the presynaptic neuron spike and measuring the TSP in the second neuron, and that's their W. And then they stimulate the pre and postsynaptic neuron in concert together in a synchronized fashion. And that leads to long-term potentiation where the synapse gets stronger. In a mammalian brain, it also gets bigger. But the strength of the synapse can be measured, again, by letting the presynaptic neuron spike and measuring the PSP in the postsynaptic. And then the change in the synaptic weight in percentage terms is shown on this plot as a function of the age of the rat from which the brain slice was prepared. And as you can see, the delta W, which is the change in the synaptic weight, reduces with the age of the rat, just like was predicted by our learning rule, which has a bigger and bigger and longer sum in the denominator. So that all makes sense.
Mitya, there is a question in the chat.
Yeah, so there is a question in the chat. How much of synaptic weights in the olfactory system are learned and how much are nature? Um, this is an excellent question, and I don't have a full answer. By nature, what is meant presumably is that the synaptic weights are encoded genetically. And we know, for example, that in the fly visual system, the synaptic circuit gets laid down even if flies are raised in complete darkness. Okay, so it doesn't depend on the presentation of visual stimuli. However, it doesn't mean that synapses are formed in an activity-independent fashion, because it is well known, for example, in mammalian visual system, that there are activity waves in the retina, calcium waves, before the eye opening. And those waves, by their correlated nature produce the activity in the ganglion cells that can wire up the visual system in the way appropriate to the images that it is yet to see. So it could be that fly has the same thing going on in the darkness. And it could be that the synaptic circuit is generated by those activity waves. Now, in the olfactory system, I'm not aware of any such experiment, so we don't really know, but our collaborators have tried to alter the synaptic weights by presenting different orders to the larva as it's being raised. And in fact, it does seem to change the synaptic weights. So we think that synaptic weights are reflective of the environment that the larva sees, but it doesn't exclude, of course, that some rough blueprint is laid down by the genes. Now, it's important, thinking about the genes, it's also important to realize that there are only 20,000 genes and there are 10 to the 15 synapses in our brain. So it's very hard to imagine that all the synaptic weights can be encoded in the genes. That's not possible. But there is some kind of rules that are encoded by the genes, and we hope that what's being encoded are the learning rules in terms of activity, which we are trying to guess here. Okay, so that's my answer at this point. We could discuss this in the end of the talk. If we feed two flies with very different orders, would their synapses be very different? We think yes, and that's what the preliminary experiments show, but that will require more work to show conclusively. Okay, so let's get back to long-term potentiation experiments. So we just showed, well, I just gave you the data that shows that synaptic plasticity goes down with age. And that's, of course, you knew that very well, because, you know, as you get older, it's harder to learn new things. Try learning a new language when you're 40, okay? It's a completely different ballgame. And, you know, here we can see that there is a mechanistic substrate that could be responsible for something like that. And that actually corresponds to the term that we have in the learning rule. So that's a nice correspondence. But it is, of course, a little bit depressing that, you know, with age, it gets harder and harder to learn. But there is another thing we can test, which is the dependence of the learning rate, not just on time, but on the cumulative activity. You see, it's the sum that grows with time, but also grows with the activity of postsynaptic neurons. Can that be tested? Well, it turns out that people have done long-term potentiation experiments in slices cut from the rats raised under normal conditions and in complete darkness. Okay, so just like those flies, they can raise rats in complete darkness. And when they prepared slices from the visual cortex of those rats and measured LTP, this is cumulative plot, they found that delta W is actually bigger for rats raised in darkness. And because the slices are cut from the visual cortex, one presumes that the cumulative activity in those neurons was less too. And so this is in agreement with our prediction that that lower activity levels correspond to preserving plasticity for a longer period of time. So that's good news, right? Because if you don't want your memory and learning ability to decay that much with age, maybe don't work that hard, okay? Skip that homework and just go take a rest. And then you will preserve the ability to learn into a few more years, okay? So again, this is nice. This experimental comparison shows that we're probably on the right track. But again, I don't want to oversell. This is biology, and there are always a lot of different data, even contradictory. So even in this same paper that showed the decay of plasticity with age, there was data from another projection shown here in white with white circles, and those synaptic weights did not show reduction in plasticity with age. Okay, is that seems like a contradiction to the theory, but actually, I want to emphasize here that finding this contradiction, just like Feynman said, is constructive because it makes us go back and rethink the objective function. And so in this particular case, it's very easy to adapt the objective function to satisfy this experimental result, because all we have to do is to think about non-stationary input statistics and adapt the objective function correspondingly. So what am I talking about here? Okay, so what I assumed tacitly so far is that all the data is generated by the same underlying vector W with some Gaussian noise around it. And that's what the PCA had to find, that W. But that is a stationarity assumption, which statisticians love, but it doesn't always, is not always satisfied in the real world. In the real world, ensembles are often non-stationary with change in W, because, you know, that rat, for example, when it was a little pup, it was searching for mother's milk. And then when it grew up, it started searching for hard food and then search for mates. So the ensembles have changed completely. And so it had to adapt to different W's. And that's why we need to have an algorithm that can learn new W's with time to adapt. And to learn this kind of algorithm, we had modified the objective function by discounting past errors in the sum. So the summation is still through the whole lifetime of the neuron or the animal, but the old errors are discounted by multiplying by this coefficient alpha less than 1, which represents the forgetting time scale, so that old mistakes don't matter anymore in the current evaluation of W, okay? And when you go through the algebra and you find the learning rule for this kind of objective, you find that the learning rate still decays with activity, but then saturates after we pass this characteristic time scale, tau, which is the log of alpha. And at that point, the learning rate stops changing. And that allows you to learn new things. And of course, on some kind of level, you also knew that semantically, because you know, babies are very good at learning new things, but they're also very good at forgetting things. So you might have met people who, or have seen kids who have learned a foreign language when they were 3, but if they were not using that language afterwards, they didn't remember it when they were 15. Okay, so this easy learning also can correspond to easy forgetting. And all of this phenomena can be captured by this theoretical framework once we add the discounting. And I think that's the real power of having a normative approach, a theory where you can rigidly connect computational tasks through objective function and optimization to the learning rule. Okay, any questions?
Yeah, excuse me, I forgot what it means to be normative. What is normative?
Sorry, good question. So normative approach or normative theory is a theory where a particular observation in biology or neuroscience is explained as an optimal solution to satisfy a certain task. Okay, so it's like optimization in physics, but it's a little bit has an additional meaning that it's an optimization of some kind of behavioral task, not just a principled objective function that is related to certain task. Okay.
So I may have missed this, but do you count, I mean, in what way, this is fascinating, but, and what I'm kind of a little bit confused is that, do you count like any, I mean, what do we count as a new input? Is it, for example, in the order, is it like exactly the same order? And if it changed entirely to, you know, either like an entirely new domain or I guess in a new domain, that neuron is not going to record it, but like a different order, does that count as a new input? I mean, like you don't put it in the same activity function, the summations of the t here, right?
Yeah, you know, it's I think it's open to interpretation as long as the it's an ensemble. You know, statistically, it's a question about the ensemble, and so every single stimulus can be anything, right? But the ensemble has to change in the sense that the Latin variable w is different.
Yeah,
that's the
yeah. I mean, yeah. I guess yeah. It to me it seems like an important question, because if we, you know, plasticity in a way is, I mean, I think there's always a trade-off between, I mean, here plasticity is good, right? But like, there's always a trade-off between plasticity and actually already having like a set of what you call like the ensemble. So these boundaries of, you know, how much whether it really reduces your learning or your plasticity given the total amount of activity it's an interesting concept that I kind of want to know more about. What do we mean exactly by activity? Because if every new things that we encode are considered as a new activity, then in some ways that that limitation that you just talk about, given this function, is can be overcome in some ways. So that's what I'm asking. Anyway, just one comment, like in infants, for example, in terms of like language learning, we do see there is very nice evidence that actually the forgetting is actually quite necessary, and this is probably one reason why infants and young children are actually better in abstracting the rule or the grammar rule in language than adults. So and we from things in my lab, actually we actually found evidence that these forgetting things are actually necessary for a lot of like abstractions at the higher level. So particularly abstractions of rules in, for example, like the rule in language, but also in other domains. So that's something that's I found this equation really interesting, and hopefully we can talk more about it. But that's what the trade-off that I mean, that the forgetting is actually necessary, and this is in fact what we believe, that why kids are actually better than adults in learning languages.
Yeah, thanks very much. This is actually very interesting. I would like to continue the conversation. And you're making a very important point that in statistical learning, you know, the goal isn't just to fit the data that you have seen. You know, it's not just the empirical loss, right? That it's important to reduce the generalization error.
Right.
on the data that you haven't seen.
Right.
And that I haven't even, it's a crucial central piece of learning theory, but I haven't even touched it because that would be implemented by a regularization term, like W squared here, which I just, for simplicity, I just didn't even put up. But that's the, mathematically, that's what will be necessary to modulate the generalization ability. And that is, in fact, related to the learning rate in the online setting. Exactly. So that's a general principle. It's very interesting. I didn't know that you could see this in human learning.
Yes, very much so.
I have another question.
Yes, please.
So just about this forgetting, is that set by the neuron itself, or what is the input? I assume different neurons would want different rates of getting information. So what is setting this alpha?
Yeah. You know, I actually don't know what the mechanism is, but as you can see in the Pu and Isaacson paper, that there are two pathways. One is lateral olfactory tract. It's the one that conducts, you know, order signals in the red from the nose to the central brain. And the association projections, these are cortical connections. And so they just have different alphas, it seems. Okay. I don't know what the sort of the molecule or the physiological mechanism that is responsible for it.
Interesting. Thanks.
Michelle, can you clarify a little bit more about this stationary and non-stationary statistics? It's basically, you're talking about this covariance matrix, right? It's going to be completely different, right? In one chunk of time and then in the other. Is that what you mean, right? Because when you're talking about covariance matrix, you're averaging over time, and you get this.
Right. So I think the sort of the formal definition of stationary non-station distribution has to do with some kind of statistics, so that, you know, if the mean is constant for the whole data set and the variance is constant for the whole data set, then they would call it stationary. But if those change, then they would call it non-stationary.
Right.
Okay.
Okay.
So those definitions, because they're statistical, they have to be done in terms of the ensemble. So, you know, every given data point can be anything, but the ensemble as a whole can have this property. And there is also a comment in the chat, but it's so said that this seems to imply a limit on the total amount of things we can learn. Yes, you know, and in fact, you know, there is a long line of work starting with former physicist whose name was Amit, and then continued by Stefano Fuzzi, looking at this kind of limit of how much memories you can store. I'm not going to talk about it, but this is something that you can look up in the literature. Any other questions?
I should add a comment, but remember also what we talk about, the generalization, right? So here it's about just a pure amount, for example, in memory, just like the amount of things that you can store. And probably nature does put a limit on it. But don't forget that that what we can learn or what we can store is not only about like particular things, like not particular memory, but we actually, a lot of our learning actually depends on our abstraction of pattern and structure. And those are, you know, we're still far away from understanding how is that encoded in neurons, but that's very important for our learning. So it might be that this is why I said this is very interesting. It might be that there is precisely the limit of the amount of stuff that we can memorize or that's embedded in this function does actually generate another function that's those are necessary limit in order to get abstraction and generalizations of patterns. So So those are very important for our learning. It's just we don't know it yet about how is that represented at all in the neuron. So don't just think about pure stuff that's getting in as the only measure of learning. So that's what I said. Like there is hope still. Yeah.
Yeah, that's very important comment, that our visual system is not like a video camera.
Yes, our visual system is not like a video camera.
Okay. All right. That's it for now with experimental tests. And again, no experiment can prove that the theory is right. Okay. An experiment can only falsify the theory, just prove it's wrong, or not falsify it. Okay. So I would say that for now, we did not falsify the theory, so we can move on. And the next thing I want to do is just to restate what we already said in a slightly different form, because that will be useful for generalizing this to multi-component PCA. So what a single neuron does, it takes this data matrix, which is composed of data vectors X that are concatenated with each other like this in time, and it decomposes that data matrix into a rank 1 product, rank 1 factorization, into synaptic weight vector and the raw vector of activities Y that grows with time. And this animation is supposed to show this online process as new data is coming in, Y keeps generating, and W was flickering, if you saw, to reflect the fact that it gets adjusted in response to the new data. Okay, and this is just going to be valuable to formulate, to reformulate this for multi-component PCA, which is what we're going to do next. So for multi-component PCA, what basically the goal here is to go beyond the single component. And that's of course what you want to do. Otherwise you just stuck with a single dimension, you don't want to be Mr. One-dimensional. And so the decomposition of the data matrix now is into this higher rank product where each column of W corresponds to synaptic weight vector for different channels, which will be represented by different neurons that will be firing a series of activity raw vectors like this. And you can represent this product as a sum of those rank 1 products that can be done by each individual neuron. And so it's natural to suspect that this can be done by a circuit like this, where the same activity data set X is streamed down to these different neurons, and it passes through the parallel synaptic vectors here and generates the output activity Ys in those neurons. So can we derive the learning rules mathematically here? Let's try to do that. Okay, so we already know the drill. We start with the objective function, which is offline, this reconstruction error minimization, except that now for each time point, you don't have a scalar Y, but you have a vector Y, each component of which is activity of an individual neuron, YT. And it's multiplied not by vector W, but by matrix W, okay, which is composed of vectors of the weights of individual neurons. But otherwise, it's the same thing. So let's try to go through with our program. The first thing we should do is to compute the optimal Y after the presentation of xt at a given step t. And you can figure out what that dynamics should be by simply taking a derivative, right? That's the gradient descent that someone mentioned before, which is what you do to minimize things. You take a derivative with respect to y. And then the cross term will give you this wx, which of course can be natively implemented by the feed-forward projections in the circuit, where synaptic weights represent the components or elements of matrix W. But then there is the second term, which came from the quartic term, which is, yt transpose w transpose w yt. This quartic term from the expansion of the square, when you differentiate with respect to y, you get this, and that will correspond to the lateral connection because it ties yt to yt. with the coefficients that are given by this product of W transpose W, which is called the Gramian. Okay, so maybe this can be done in the circuit that I showed, but the problem arises when we try to derive learning rules. So to derive learning rules, we take a derivative of the same objective function, but now with respect to W. And to demonstrate the problem, I wrote down the solution for the feed-forward synapse connecting neurons I and J, say 3 and 2, okay? So then for that synapse here, we take the old value, we add the product of the postsynaptic activity times presynaptic activity, that's fine. But then we have to subtract this horrible sum. The reason I say it's horrible is because it invokes the activities of all the other neurons in the system, YK. And how would this poor little neuron, which is less than a micron in size, embedded in this neuropil, how would it know about the activity of this neuron or any other neuron in this layer? That's just physically impossible. And that's an example of a non-local learning rule, exactly the kind of learning rule that I warned you against in before. And so this kind of learning rules, just like the backpropagation algorithm, cannot be implemented in the brain with known biological or physical mechanisms. And so we run into a problem with implementing just a simple principal component analysis for the case of multiple component with multiple neurons. And the problem only gets worse when we start thinking about the lateral connections, because they need to be adjusted as well. But there isn't even a learning rule for them. The learning rule is for W. And then once we adjust W, then we have to compute the Gramian. How would neurons ever do that? That's beyond me. So that's why there was not a biologically plausible implementation of multi-component PCA. Is this clear? Okay, so given this situation, the scientific community took two paths. There is a whole bunch of papers where people said, oh, okay, well, these learning rules are not biological, too bad, but these are still interesting machine learning algorithms, so we'll keep using them. And you know, they may not be implemented in the brain, but they're still interesting algorithms. Then there was another group of papers that said, well, we really can only use those learning rules that could be implemented biologically in the brain. And so this would be the local learning rules. So they fudged those learning rules a little bit to get rid of this kind of sums and have learning rules that kind of work. And then they showed numerically that they did something useful. But because those learning rules were not derived from a principal objective function, it was not the output of a normative approach, they didn't really know what kind of output the network will give to any random data set. And so every time you would have to simulate to know what the output would be. So the predictive power of that kind of theory is not that big. And that's why we were dissatisfied with both kinds of choices. And we were determined to find an approach that is both normative, where we can derive learning rule from the principal objective function, and the learning rules are local, and that is biologically plausible. And following Feynman recommendation of what we did when we encountered this disagreement with experiment, we went back to the optimization problem and guessed a new objective function that was close enough in the sense that for a single neuron, it produced the same learning rule. But for multiple neurons, it gave a different learning rule so that we could have a local biologically plausible learning rule. So this is the subject, this approach, which we call the similarity learning, similarity matching approach, will be the subject of my second lecture. But before I conclude, I should also mention that I'm kind of simplifying things here a little bit. It is also true that this problem with non-locality of learning rules was realized very early on. And for example, Olshausen and Field proposed a different circuit which circumvents that problem. Sorry, my fault. That circumvents this problem by building a two-layer network. And this two-layer network actually can be derived from this same objective function that I showed you with it gets local learning rules. And we were at some point fans of this two-layer network, but the more we tried to compare it with experiment, the more we realized that it didn't really match. And so that's why we abandoned that approach, just like Feynman quote said, and try to develop this similarity matching approach. So I think this is all I wanted to say. And so I would, yeah, that's it. And I'm open to discussion. And if you have any questions, now is the time.
All right, thank you, Mitya. So it's questions time for the students to ask any questions that you want. And I don't have to moderate. You can just go ahead and unmute your microphone if you have any questions and just ask Mitya directly.
I would like to ask, what is the first nonlinearity that one would want to add to the model? Based on, I don't know.
Yes, so that's the good question. I think the answer will depend on whom you ask. What we did, we added a rectification nonlinearity. So we turned neurons into rectifying linear units. And in the second lecture, you will see how and why we did it. But that was our approach. But someone else could come up with a different nonlinearity.
What is a rectificator? You said it earlier on the talk.
So you get the inputs, you compute a weighted sum of those inputs. If that sum is greater than 0, you output the sum. If that sum is below 0, you output 0.
Perfect, thank you.
So I have a relatively general question. So in general, you're talking about this normative framework, framing each question into an optimization problem. And I just wondering why it's so important to have this normative framework or normative thinking. Why we're like trying to find this kind of like explanation of principles in the brain. And biological process seems to be too complicated, and like since that doesn't like look like physics, right? So like, do you think this kind of normative approach would be also applicable to like for us to really understand the brain and a lot of the biological processes?
That's a good question, and it actually has been debated in the biological community. You know, I think it's semantics to a degree, because you know, we are not really trying to prove here that the brain is optimal in some sense, and we are not even testing whether the brain is optimal. What we're doing is we're using optimality as a tool to understand the brain. we are trying to make sense of why things are the way they are. And usually that implies a deeper level of understanding than just answering how questions and then just describing mechanisms. And to answer the why question, optimization is like a natural, it seems like a natural approach, because we know that evolution optimizes fitness. So we know that biology is aware of the optimization. Of course, there is a long way from optimizing fitness to optimizing the reconstruction error. And that's a leap of faith. But I would say that it is, in some sense, reflective of our mind, our meaning scientist mind, not just the brain that we are studying, that optimization becomes a useful tool.
Perhaps another consideration on this question, it is a very interesting question, is that it's kind of hard to imagine what are the alternative functions that you might want to do. So I mean, optimization is pretty clear to us. That's one thing that could possibly actually as a force in nature that works. And then if we ask ourselves, like, what are the other alternative functions? Like, what our non-optimization looks like, then it becomes very difficult, you know, so it's kind of becomes very undefined. So maybe that's another way of thinking, you know, we always consider alternative models, right? Alternative possibilities about how nature works. So same thing even in biology, like it's sort of hard, and maybe Mitya can tell us more about what are the possible alternatives about how might biology work, if not optimization. What would that be called? Like so that might be another reason for, at least as a, you know, a first try, like optimization sounds like a good tool. But, yeah.
Yeah, that's a great point. There isn't an alternative.
Yeah, and it's like the alternatives are missing. You know, it's like as scientists we have to try alternatives, right? So
perhaps the alternative could be like you have many different, like in physics, right? So we have like very fundamental laws, fundamental theory like general relativity and like quantum mechanics. But on the practical level, we have like many different practical theory on the very like top surface, doesn't have deep mechanistic explanations, but can be useful in many engineering fields, right? So perhaps that could be one alternative, I guess. I don't know. So what do you think?
I don't know. I mean, engineering is all about optimization.
yeah, I was about to say, I think.
The way I see it.
Yes, it's
You can argue about, yeah, please.
No, go ahead, Mitya. Yeah.
You can argue about what your objective is. Are you minimizing the cost or the time or the material amount or something else? But in the end, it's all cast in terms of optimization and engineering. Okay, while people are thinking about questions, I have one more slide that maybe is worth showing, which is a comparison of you know, artificial neural networks and natural ones. And it's you know, one important thing that we didn't talk much about is that there are different learning settings. There is so-called supervised setting, where the network has to classify the data, say cats and dogs. And then when it comes up with the label, the trainer, the teacher, tells the network what the correct answer is. And that creates the error, which can be used to adjust the weights in the network by this deep propagation algorithm using non-local learning rules. But the important thing is that there is a true ground truth, a true label that adjust the data. In biology, although this also exists, most of the learning is unsupervised, where the ground truth labels are not available. And most of the labels actually is done on very few labeled examples, or very few examples, period, that people can learn. They call it like one-shot or zero-shot learning. And that's very hard to implement in this sort of artificial network that is designed for supervised learning and require hundreds of thousands of labeled examples. And then another is this local learning rules that we talked about. And yeah, and then, okay, it's a joke.
So my question about this slide is, so people has been talking about one-shot, a few-shot learning, and it seems very hard to be implemented in biological neural circuits, right? So I just wondering, like, is the kind of one-shot learning or few-shot learning really learning, or it just like we better frame it as inference?
Well, I mean, okay, I'm not an expert on this, but my understanding is that to do a zero-shot or one-shot learning, you can see as many unlabeled examples as you want. So you can do unsupervised learning first, and then once you learn some kind of structure in the data set, then someone shows you a single label, and then all of a sudden you know a lot, and you can, you know, you can classify future examples well.
Yeah.
I don't know if someone.
Yeah, I just thinking. Yeah, people has been talking about one-shot learning is important, but like I don't actually think that's learning, right? Because once you get to the point, you can do one-shot learning, and most of the learning is done, right? So this is at least my understanding, but I don't know if that's
most of the learning happens before the shot. Yes.
Yeah. Yeah.
Exactly.
Yeah.
I think I mean I wouldn't say that inference is not learning. I mean, inference is a very important part of learning. I think one of the basic definitions of learning is that can you make sense of the new data that you get in, right? So it's the process about the making sense, whether the making sense is using an underlying structure, or what is the process of that making sense, and what kind of ingredients, so to speak, that you need in order to make sense. So I think that can be that is kind of like that is something that can be debated about that. But as long as you are able to make sense of a new data, of something that you never seen before, then I would say that that's learning. Obviously in cognitive science there is a lot of debate about well, there's some debate now, it kind of dies down a little bit about there's no such thing as learning at all, but that's a Fodorian he's a philosopher. There's a Fodorian argument that there's no such thing as learning, everything is there from the very beginning. But I think we have it's a bit untenable at this point. So I think you should think about learning as how do you make sense of the data, and it's the process of making sense that we're still trying to figure out, like in what way do you make sense? Do you make sense based on the available structure? Do you make sense based on a similarity matching to the previous data or item that looks exactly like that? Or do you actually there's even evidence now, you make sense of it because of dissimilarity, so the opposite of similarity. So there are many, and those are the many ways of making sense of the data that I would call like learning. So Mitya, I would like to ask whether the linear, the optimizing linear, decoding that you've done here does actually take into is actually able to accommodate these this unsupervised learning that we really see in biology. So I keep coming back to the questions about how do the neurons, it's about the input, right? So every time there's an input coming in, of course, we know that there's no homunculus, as you mentioned in the beginning, but what makes the neurons want to do the linear decoding to particular input, and sometimes do not want to do that linear decoding to that particular input?
I actually don't know of any strong argument other than convenience.
Okay.
And as I was saying in the beginning, if your representation is divergent, so you expand the dimensionality of your representation, like from the retina to the visual cortex,
right.
There are many more dimensions, then the encoding process could be nonlinear. No, we know it's nonlinear, right? They're on
right.
and off cells and so on. Okay, so the encoding process, it's nonlinear, but this high dimensional representation could very well be linearly decoded, right? Because that's the nature of high dimensions, that you can go down now and reduce the dimensionality by a linear transformation. Of course, that's not what this representation is trying to achieve.
Right.
I think the goal is, which is closer to what really probably happens in the brain, is that the classification, the behavioral output can be decoded linearly. That's probably what is really going on. But people have had a lot of difficulty thinking about the motor and the decision-making end. And so they just said, okay, let's do linear decoding and go back to the sensory. But in reality, you want to go forward, not backward, and have linear decision-making once this representation has been made. And in the fly, where the circuit has been mapped out, that seems what is happening, right? There is a huge divergence from that olfactory circuit that I showed to the canyon cells in the mushroom body, where you go from 50 to 5,000 dimensions, and then from 5,000 you go to 30 neurons that determine the behavior. So that transformation is probably linear.
Yeah.
Can I ask a sort of general question? Yeah, so today's in your lecture, you are mainly focused on the keywords of biological plausible. And I have noticed that currently many works are trying to apply those so-called neuromorphic computing to apply these biological features that we observed in our brain or in biological brain into the artificial neural network to improve this, I don't know, to improve performance or sort of improve this energy efficiency of the artificial neural network. But it seems not, so far did not get very promising result. So my question is, do you think this kind of approach is sort of necessary, or so far what kind of parts are we missing when we are doing such kind of digging?
Are you asking about about sort of artificial intelligence things?
Yeah, how can we just appropriately to apply those biological constraints to this artificial neural network to sort of improve the performance or to improve the energy efficiency or some other kind of things to improve the artificial neural network?
So this is a very interesting question. And I have to admit that initially when we started working in this direction, our goal was to understand the brain. Because we thought that, if we're solving some machine learning task, like dimensionality reduction or clustering or whatever, and we introduce additional constraints like local learning rules, then we're going to be at a handicap relative to the general purpose machine learning algorithms. So our goal was to understand how the brain works. But over the years of doing this many, many times and revising our objective functions, going back to biology, revising the objective function again, we actually realized that those constraints may not induce a handicap. And in fact, they may actually help you to develop in the direction of the general artificial intelligence, which is something where, so the machine learning community tries to go, because, it kind of, it's a blessing in disguise, right? That those constraints are not like roadblocks that prevent you moving forward, but they're more like guardrails that tell you in which direction to go. Because we know for a fact that there are things that the human brain can do that artificial networks can't. So there is some secret sauce in there, okay? And that has to satisfy the constraints. So we know that the solution to that sort of general intelligence can be done under those constraints. And this reduces the space we need to search in. So now we're kind of more optimistic about our ability to actually come up with competitive general purpose algorithms.
Mm-hmm.
Okay.
I also just want to add here, Kai. So also in my own works, I work with young infants, and so we try to understand how does the mind work from the very beginning. And in my work, I work together a lot with AI people, because now there is a move in AI to make machine learning that can make sense. And I think one of the interesting, like very different approaches that we try to bring together is that in my work, we always try to understand constraints. So if you actually try to if you read like things, the most advances that we have understood in cognitive science are what are the biases and what are the constraints that the mind imposes, such that the learning is so amazing. And when I work with AI people, I think one thing that's very interesting to me is like, in AI is like more is always more. But in human is actually, it's how do we make from less is more. So for example, the forgetting that Mitya mentioned, that's why I think that function is so interesting, is that that's something that's really important. So for now, like right now, we actually don't know. Actually, there's no attempt in AI to in deep convolutional network to actually forget. And we don't actually know what it means, but I think it's actually really important, because this constraint of forgetting is something that we see very, very importantly underlies the ability to abstract pattern. And as you know, one of the actual difficulties in AI is to actually abstract relations and patterns. Like it's very difficult to learn, even for deep mind, to learn simple topography like top and bottom. And humans, young humans can do this from very easily, but this is still very difficult. And I think it's something to do to understand top and bottom. You actually have to forget what the actual things that you see. You have to forget that it's about an apple, but it's about the top and bottom relation. So that's why it's so I think these constraints are actually really interesting, and I foresee that in the near future. I think this is something a big important direction in AI and in modeling in AI to actually really look at the constraints. So not more and more, but actually going a different direction. Less is more.
Yeah. Thanks for the comment. Yeah. One more thing that I've noticed that you have mentioned about this general framework of computing, or sort of general computation to do multiple tasks, multi types of tasks for using the same architecture or using the same network. This is what our human brain are capable of, but for artificial intelligence, for artificial neural network, it's hard to do that. So seems like our future goal is trying to crack this general framework of computation, but so far it is some works have already done some trials or in such kind of area, but I found it's very difficult to characterize or to sort of calibrate this ability of general computation for example, for a specific framework, because nowadays people are using sort of data set in commonly used in AI, such as MNIST or CFAR or ImageNet to characterize their new network to do different classification task or some sort of thing, but it is still quite far from what we have just discussed about this general computation or sort of general framework of computation. So I think maybe it is necessary to sort of came up a standards or came up a frame of calibration for this general capability of computation, so that we can actually when we came up a new algorithm, so that we can calibrate the ability compare with this old framework, so that we can say that this is this new framework or this new algorithm is worth to develop, right? I don't know, but just maybe just comment, yeah.
Thank you. Okay, so there is also a comment in the chat, and I should probably let you go because it's already 10:08. So the goal is to utilize how brain works to maximize the computing efficiency, accuracy, and less focus on understanding how the brain works. With this biological constraint still can be seen as some indicators for directions we should search for or acting more like roadblocks. In other way, would the imitating the biological mechanism be the most efficient way to do computation? Okay, so I don't know. So my primary goal is to understand how the brain does things. I think that what we learn will be useful because the brain can do things that computers cannot at the moment. But I don't want to pretend like I know how to how to build the most efficient machine learning algorithms.
If there are no further questions, because we are ran out for time, and I'm sure Mitya is quite exhausted already. And thank you so much, Mitya. It's such an excellent talk. Some people actually, I don't know if you noticed that a few students actually come again after listening to your yesterday's talk. So it's also great. All right, thanks, Mitya, and we'll look forward to the second lecture. So in the advanced in advanced section, and I think for the students, we're staying over for the group 4 student presentation. Thanks so much, Mitya.
Thank you. I had a blast.
Thank you so much, Mitya.
Bye.
Bye.